My name is Michael Veronesi. I'm both an MD and a PhD. So I did a PhD in neuroscience back between 2004 and 2008. And then I earned my MD back in 2010. I went on to do a neurosurgical internship at the University of Virginia. And then after that, did a radiology residency and then a neuroradiology fellowship. So I joined the faculty at the Indiana University in 2017, and then most recently joined University of Wisconsin here in June of 2023. So I'm still getting things up and running here at Wisconsin.
Cool. What is your broad research area?
Clinically, I'm a neuroradiologist, and then I have expertise in training all the way from bench to bedside. So um, mostly what I do is study drug delivery in combination with imaging. So the concept of Theranostics, also utilizing advanced MR and PET, so magnetic resonance imaging, and positron emission tomography. So, and then this is specific in the last 4 or 5 years to brain tumors. And within the brain tumor realm, mostly I study the primary brain gliomas, glioblastoma, but I also have research projects in brain metastases as well. So, but everything's within the central nervous system, and then everything's within a neural focus.
How did kind of your PhD work and then like your clinical training
Mhm.
lead you to studying glioblastoma?
I actually started out in epilepsy, so I study in neuroscience mechanisms of epilepsy, and then we developed a very unique route of delivery for drugs through the nasal route from nose to brain. We worked with nanoparticles, and then had a really potent anticonvulsant compound that actually got efficacy in an animal model. And that led to the question of studying the nose to brain route or other routes to get around the blood-brain barrier. So, which is, the number one issue probably for developing drugs against any kind of brain disease. And so it kind of ends up being, well, whether it's epilepsy or brain tumors or Alzheimer's, it's really the same, very similar question of how do we get drugs to the place they need to go in order to treat patients. So what I did when I was in training was start to study those mechanisms of transport. And I did that with some grants that looked at from a hypothesis-driven approach of using advanced imaging. So that's where the imaging started to come into play after the PhD in medical school during the radiology residency. And so we got some interesting results that we published, and then that led to further grants and things like that. But um, from an ability to overcome the blood-brain barrier, the brain tumor realm is a very good realm to study. There's very good animal models. They have a lot of different possible drug candidates, and so the field is very ripe in terms of drug delivery for brain tumors. But also the imaging is extremely good at looking at brain tumors. Sometimes it can be hard for looking at some mechanisms in other diseases that may be a little bit more diffuse within the brain. But brain tumors, as you know, can be pretty localizable on the imaging, and you can see if that drug that you're testing with is working. And so what I developed was more in vivo systems for studying the effects of brain drugs in the context of brain tumors. And so I still do the nasal delivery. I also do intravenous and oral delivery of drugs in the animal model. So that's on the preclinical side. On the clinical research side, so because as I'm a neuroradiology clinician, but I also have half of my time I'm a physician scientist. So on that side, I work to translate those novel therapies and imaging devices into the clinic. So anyone who works with me would have the opportunity to understand all the way from a mechanistic approach to disease and overcoming barriers to drug delivery to the brain, all the way up to seeing how it's working in patients, both on the therapy and the imaging side. So that's where those advanced MR and PET imaging techniques come in. And so what I'm looking for is someone who's very excited and passionate about developing novel ways of using the imaging to look at brain tumors. So the two specific examples of the research that I do on the PET side, I utilize amino acid PET, and that's a compound called 18F fluoroethyltyrosine. I also study 18F flucyclovine. And then finally, I'm with a group here at University of Wisconsin who's studying fibroblast activating protein inhibitor, which is another very exciting PET compound for possibly looking at brain tumors. And so we need someone very excited to help us develop that. On the MR side, we have several different advanced MR techniques and sequences. The one that I'm really studying is called chemical exchange saturation transfer, or MR-CEST. Another word that's commonly used is amide proton transfer, or APT-CEST. And these are endogenous ways of looking at brain tumors that highlight the protein that's being produced within the cellular milieu of the brain tumor environment. So when these are elevated, the imaging, that particular imaging, can help pick that signal up. And it's exciting because it's a standard MR technique, so it can be used as part of your brain tumor sequence. It's close to clinical care, but it needs further development. It needs further standardization. We need new ways of making the signal be more present and visible. So all of those together will help this entire field, this what we call PET-MR field of theranostics. So those are broader category terms for this entire research program.
Could you tell me a little bit more about, on the clinical research side, how you're sort of identifying the different populations that you want to work with, and also more so of like what you're doing in terms of the different studies that you're working on right now?
We have we have relationships with neuro-oncologists, neurosurgeons, radiation oncologists. And then what will happen is we'll have patients that will be being treated as, with certain drugs or compounds, and then they need to be followed with imaging. And so what we do in these conferences is we get together as we talk about, well, what's working, what's not working, and how can we push the field further to develop some of these things that are kind of at the cutting edge, but are not quite ready for prime time for clinical care. So, and then how do we develop investigational new drug applications and IRBs to be able to test these compounds out in patients who are willing to be part of this study, and may or may not derive benefit from them themselves, but obviously for the greater good of increasing the science. So that's where the clinical realm sits. Is we've tried something out preclinically, or it's been being used in clinical research for a while now, and now they need someone who can sort of bridge the gap between clinical and research. And then within that domain, you have certain regulatory bodies that are going to require certain things, and then certain things have to be done a certain way. And so, but the opportunity to test those novel drugs or imaging devices, that's kind of the realm where I sit in the position that I have. So anybody that would join my lab would understand, this signal transduction cascade could be the mechanism of the way this drug could treat this tumor, all the way up to what's going on in the patient realm when we do this advanced imaging, and is that drug helping that patient survive longer?
Would you be able to tell me about your clinical research split and kind of what that looks
Mhm.
like for the rest of your lab as well?
Sure. I'm really happy that I have a nice layout for my clinical and research. So the typical clinician will get somewhere between 20% time towards academics and research if you're in academia, all the way up to actually 80% research and academics if you have very big grants and you have a large research team. I'm somewhere in the middle where I'm 50-50. So 50% of my time I'm in the clinic, and 50% of the time I run this lab that I have in this research side of things. Within the research that I do, 25% roughly is, 50% is established towards the clinical research domain, and then 50% towards thinking about, novel things that are not quite ready for the clinic, and very open for further development in the experimental side. So that's kind of the layout that I have. But the nice thing is that for 75% of that, I'm really in the clinical care realm. So folks can get a hold of me on the clinical side 75% of the time when we have interesting things that are going on for patient care. And so being integrated into that multidisciplinary clinical team is extremely important for furthering some of the questions, answers to questions that we have on some of these research studies.
What are some of the things that your lab has found in the past couple of years that are really exciting?
Yeah, so I have fortunately, I've been fortunate enough to publish very recently in both the clinical and the preclinical realms. So nicely enough, I have an easy way to refer someone to very recent publications that I have. On the clinical research side, we implemented the amino acid PET into clinical care using simultaneous PET-MR. So with the 18F-fluoroethyltyrosine, it had not been done, or at least published in an organized manner over a larger number of patients spanning glioma and brain metastases in the United States. It's been done very well in other countries over the last 20 years, but the United States hasn't really done that in the context of clinical care. There are a few places that are doing it, but so we were excited to be able to publish this to say, we've done this, and it does work, and it does work very nicely in patients in the United States. And so it's sort of a paradigm shift towards thinking about, most places only do brain MRI, with a few places doing PET MRI. And we've been able to produce an example to show that the amino acid PET MRI for brain tumor imaging is really, really helpful and efficient. So that is leading to some efforts by pharma to think about, should this become FDA approved? What do we need to do to develop this further in order to make it more widespread in the United States and help patients, right? So that was published in the Journal of Nuclear Medicine. It's a part of an 80-patient study that we did to study all forms of the management, but mostly within the post-treatment phase, where the MRI changes start to kind of confuse people as to really what's going on with the treatment. And then this helps clarify that. On the preclinical side, we published in oncology and basically used an animal model, an animal rat model, applied the same imaging techniques of the advanced FET, PET, and also now integrated the MR-CEST into the MRI side of that animal model. So we delivered drugs to the animal model, and we imaged with the advanced PET-MR imaging. And so we published that in Frontiers in Oncology. So that's available too for anyone to have a look, that can kind of demonstrate that that's the type of research that we do on the preclinical side.
Would you be able to tell me more about your research team and who's in your lab?
Since I just moved from a different institution, I'm building my lab. I have collaborators that I work with in multiple various different projects. I have a job posting for both a lab manager and for a postdoctoral fellow, or what we call at University of Wisconsin a research associate. So both of those would be integral to performing both the clinical research and the preclinical research. But a nice thing about the University of Wisconsin is there are a lot of cores and a lot of already well-established other investigators. So this work is ongoing. But yeah, definitely I would benefit from a postdoctoral fellow who would be really excited to be involved in this research.
How would you describe your mentoring style?
I'm pretty. I think if you talk to students and postdocs and trainees who've been with me before, I am more of, hey, what do you want to do, and then let's help you get there. I'm a little more hands off, and I think initially, folks might say, well, I need some direction. But then as you get into it and they realize they can chart their own course on a lot of things, I think people really grow in that way, with being able to try to answer the question themselves first, come up with a solution in a novel way first, and then when they're really struggling, they can come to me, and then we sit down and we look at it, we figure it out. So my research has always been, as a clinician, I don't have 100% time in the lab, so
Mhm.
the person who joins with me will have to kind of be a go-getter and someone who's not afraid to try new things, kind of go for that, go for the gold, and see if you can come up with the answer. So I think I've worked with students for a number of years now. I also train medical students, residents, and fellows. Also help mentor junior faculty, and so my hats that I play is pretty varied. So I'll have students that I'm helping or involved with, whether on the research side or the clinical side, from college all the way up to faculty. So I think that's kind of the style that I have is, you and I are equals. We're working on this together. There's no I'm above you telling you what to do. There's more like, you have my respect already because of the hard work that you're doing and what got you here now. But so let's figure out how to help other people and help patients, and let's try to help make incremental steps towards the process of curing a disease, or at least make helping patients survive longer, and improving various aspects, whether that's therapy or imaging.
What are some of the most important skills that you think trainees develop in your lab, both on like the preclinical as well as the clinical side?
Yeah, great question. I think because I span from the bench side to the bed, I don't have a requirement that someone who joins my lab know all of that. That's a lot of different skills and techniques. Some very specialized on the animal surgery side, all the way up to coding and programming on the advanced imaging side with humans and with patients. Um, the particular posting that I have out there really focuses on this clinical research realm, with a small portion in terms of the postdoc with on the preclinical side. That's predominantly because we're really in an exciting time where we have huge potential to get this into the clinic, the imaging aspect, and some of the theranostics that we're doing to really change how we take care of patients. And so I think that's what's really exciting. And that's where one of the reasons I was brought to the University of Wisconsin to focus on. So that's the area where folks with that particular expertise, so they would understand the basics of MR physics. They would have kind of a sort of an engineering, technical mindset, a coding mindset, understand the basics of artificial intelligence and radiomics, things like that, where that's where the field is headed. That's what the future holds. And, that's really where we're positioned. So that's where that postdoctoral fellow would fulfill that.
How do you see your team and lab growing in the next couple of years?
We have multiple areas where, as a university, we're growing, as a school of medicine, as a department, as a section in neuroradiology. And so, there's increased clinical demands because patients are getting their care more. And so what we have a need for is to more quickly come up with the answer that clinicians need in order to decide what's best for the patient. So the areas that my lab probably will grow will be in those, collaborative efforts that are, multi-investigator, that are seeking to solve these, problems of these larger, scaling issues that we need to have in order to get, more efficiency and take better care of patients. So I think the questions are going to be, okay, yeah, we could do this, but now how do we get it so we can do it quickly and get it into the context of the fact that we need a fast turnaround clinically. So those are the challenges, and that's the area where we need to grow.
If you could give one piece of advice to someone joining your lab, what would it be?
It's going to be both exciting and at times, a little scary, because we do a little bit more high risk work because we're going for important solutions. So this would not be a lab where, you're going to get your set of instructions already laid out for you and you're going to follow a recipe. This is really a domain that even those of us who are experts in the area are still trying to figure it out. So that's why it's exciting, because you can really quickly get involved in things that are groundbreaking, new, novel, going to shape patient care. But at the same time, as artificial intelligence, and larger scale therapy trials, those are all things we're still trying to figure out. Even the best, the most renowned experts are trying to say to themselves, well, there's a problem here, and this is what's keeping us from translating it into patient care realm, not just locally, but across the nation or across the world. So that's someone who is joining the lab, likes to take risks, likes to be on the cutting edge, a little bit on the edge of the comfort zone, where maybe this is not your area of expertise, but you're bringing your area of expertise in addition to learning some other really radical or really interesting techniques that will sort of allow the postdoctoral fellow to be more well-rounded and more familiar with areas that they might not otherwise get exposure to.
Thank you so much for sharing that. And I was wondering if you had anything else that you would like to sort of speak about your lab or about your research program or anything that we haven't covered so far.
No, I feel pretty good about it. I obviously, we're accepting applications anytime, and if the candidacy looks pretty good, then we'll set up an interview. I really am, to be honest, focusing on this PET and MR concept. If a postdoctoral fellow comes in and says, "Well, I've done mostly MR to date, and I feel like I could pick up the PET part of the research," and vice versa. If they come in and say, "Well, I've got really good PET understanding, imaging wise, and but I could totally pick up the skills for the MRSS part or things like that." So coming in with at least one strength of, say, maybe 3 or 4, and then having the confidence and the capacity to learn the other 2 or 3 would be someone who would do really well.